In this experiment, you will use functional group tests to detect the presence of the phenol functional group. We will also use a pH indicator to determine whether acidic or basic functional groups are present, and we will use an iodine indicator to detect the presence of the polysaccharide starch in your materials. First, you will test various materials using solution tests. You should put a small amount of each material into a test tube. Use an amount that is about the size of a grain of rice. You will then add water to your tubes to dissolve the solid component. Then you will add your color indicators. For example, if you are testing for starch, you will add the iodine reagent. After completing the solution tests, you will repeat these tests on paper. Each group will start with 4 of the blank paper analytical devices. The blank pads will already be cut for you. You will add your reagents to the top of the pad lanes. These lanes are labeled A through F at the top of the card. Add 1 drop of iron 3 chloride to one lane, one drop of universal pH indicator to another lane, and 1 drop of iodine solution to the third lane. Before doing anything else, make sure to label your pads with the component that you plan on testing on each of the pads. You should have 1 labeled salicylic acid, 1 labeled synthesized aspirin, a 3rd labeled aspirin tablet, and a 4th labeled Tums. The next step is to swipe the materials to be tested across the swipe lane. This lane is indicated with either a green block or a black arrow, about 1/3 of the way from the top of the lanes. Place your powder across the pad lanes using a wooden spatula. Then use the spatula and use some force to rub the powder well into each lane. Push hard to do this. Dispose of any excess powder in the wastebasket. In order to apply the aspirin tablet to the pad, you need to use a mortar and pestle. First, break the tablet, then grind it into a fine powder. Once the aspirin tablet is pulverized, you can apply it to the card using the wooden spatula. Make sure you break the tip of your wooden spatula off so that you don't contaminate your aspirin with other components from previous pad applications. Between applying each substance, break the tip off of the wooden spatula so that you're using a clean spatula each time. To develop the pads, prepare a beaker with a small amount of water in the bottom. The water should be very shallow so that when you place the bottom of the pad in the water does not go above the blue line. The water will be drawn up the pad. When the water reaches the top of the pad, you should remove it and lay it flat on the bench top. Each pad will take approximately 3 or 4 minutes to develop. You can develop 1 pad at a time, or you may be able to fit 2 pads in 1 beaker. If you attempt this, beware that it is at your own risk. Do not let those pads touch one another. Develop each of your 4 pads in this way. When all 4 pads have been developed, you should take a photograph of the pads with your camera phone so that you can refer to this photo later. Now you should record all of your observations in your data sheet and interpret what the results mean. Then, before moving on to the next part of the lab, make sure you wipe off your benchtop. In the next part of the lab, you will be assigned a drug. The pad you will use for this part of the lab will already have chemicals loaded onto it, and it will have lanes labeled A through L. Some of these lanes contain the test that you just used in the previous experiment. There is a very small amount of powder in each capsule. Make sure you tap all of the powder down to one side before you open the capsule. Carefully open the capsule and tap the powder onto the pad along the swipe lane. Get as much of the powder onto the pad as you can. Be very careful not to spill any of the powder onto the bench top. Then use a clean spatula tip to rub the powder onto the swipe lane. Use force and really grind that powder into the pad paper. Make sure that the powder is evenly dispersed over all of the lanes. Tap off any excess powder. Make sure you label your pad using a pencil. And then you can put your pad into the developing chamber with a small amount of water at the bottom. Make sure the water is shallow so that it doesn't go above the blue line on the pad. While your pad develops, you can prepare for the next step. Get the worksheet which contains the pad use instructions and also has a picture that says place pad here. On this piece of paper, you should write the make and model of your camera phone and the type of drug that you are testing. The pills that you use should have a red or blue spot, so please write the color of this spot as well as the name of the medicine. When the water reaches the top of the lanes, your pad is fully developed. You should take it out of the developing chamber and put it onto the piece of paper that says place developed pad here. Now set a timer for 3 minutes. As soon as 3 minutes is up, then you should take a photo of the pad. Make sure that your photo captures both the pad and the information next to the pad which you wrote about your camera phone and about the drug name and color. Then please email your photograph to the email address listed on the information page right above your developed pad. By sending your photo to this email address, you are helping the PADS team at St. Mary's College and at University of Notre Dame to advance their research. So thank you for your help. Now use this photo to answer the questions in your post lab and see if you can determine what's in that pill that you tested.